intense. That's how it goes.
3 minutes was shorter than you think it is.
3 minutes, it's enough to say what's the purpose, what's the problem you want to solve, how you're going to do it, and what does it take.
What you're looking at right now is the first schematic for a cure for the deadliest infectious disease known to humanity.
I talk a lot, so at different presentations I see myself running beyond time. So the participation has helped me learn how to carve my story and then present it in a pitch form in a very short time.
Thank you very much.
I really fell in love with this event because it brings together all the great minds on one stage.
You need to be also out of the box in order to share, to feed your own research with the contact with the others.
I hope that the future sees more collaborative efforts like this, where people come together, bring in different strengths and fusing those together to solve complex problems. And science will be key to that journey.
Good morning and welcome to Falling Walls Lab Queensland 2021. Well, we hope to have this event in person this year. Given in these uncertain times and lockdowns, we had to pivot towards the online event. Thank you for all joining us to this event wherever you are. My name is Hans Bachor from the Australian Academy of Science, and I will guide you through today's program. Before we begin, I would like to acknowledge the traditional owners and the custodians of the lands where UQ stands, one of the key hosts of this event. We respect to the elders, past and present and emerging, who continue cultural and spiritual connections to our country. Let's recognize the important role Aboriginal and Torres Strait Islanders have played and continue to play within our community and their valuable contributions to Australia and to the global society. I'm thrilled to be the MC for you this year again and this annual event, and look forward to seeing the various ideas our young researchers will present and their ways how they want to break down the walls that still limit our societies. Queensland's Falling Walls Lab is a partnership between Study Queensland, the Falling Walls Foundation, the Embassy of the Federal Republic of Germany, German Consulate in Brisbane, the Australian Academy of Science, and the University of Queensland. Falling Walls is an initiative created in Berlin, Germany, the place where in 1990 the wall fell down and many great ideas were turned into action for a peaceful world, for a safe, prosperous, and healthy world. Now, Falling Walls Labs brings together young scientists, engineers, professionals from across the globe to present their vision on how to make the world an even better place. As you would expect, there are thousands of ideas from across the globe, and many of them have nominated through about 70 parallel competitions in different countries. Now, the key event of Falling Walls will be in November in Berlin, when 100 presenters will address an audience of decision makers, people who can turn your ideas into action. Each presenter anywhere in the world has just 3 minutes to make their case. to explain the wall that's in the way of progress and to explain how this can be done. So who will be the key influences of the future? From Australia, we will send a team selected in a national event of the Australian Academy of Science. This year, we're doing that for the 5th time. In September, a national jury will find the most impressive visions from our country. 10 young colleagues will compete for a place in the Australia team. And amongst them will be 3 right here from Queensland. Now, before I introduce the finalists and the outstanding jury that is with us today, it is my pleasure to introduce Dr. Jessica Gallagher, Pro-Vice Chancellor of Global Engagement and Entrepreneurship at the University of Queensland, to give an official welcome. Welcome, Jessica.
Thank you so much, Hans. Good morning, everyone. It's my great pleasure to welcome you to the Falling Walls Lab Queensland. I'd also like to begin by acknowledging the traditional owners and their custodianships of the lands where we meet today. I pay my respects to their ancestors and their descendants as we walk together on the path to reconciliation. We are delighted that UQ is hosting the Falling Walls Lab Queensland for the first time this year. And I'd like to say a special thank you to Study Queensland for partnering with us and supporting this initiative. Also, thank you to Professor Hans Bachor and the Australian Academy of Science, and to our Honorary Consul for Germany in Brisbane, Professor Michael Rosenmann, for continuing to put your generous support behind some of our brightest minds here in Queensland. Although we're not able to be together in person today, I'm absolutely delighted that we're still able to press on with this event. I'm thankful that today's event is filled with innovators who are no strangers to being agile, adaptive, and resilient to sudden changes. UQ has been involved with Falling Walls since 2016 and will continue to throw our support behind an initiative that gives innovators a rare audience to which they dare to create a better world through science and social change. Why? Because it allows us to invest in our best assets, you, our brightest, most innovative future leaders. It is UQ's vision to provide of our efforts to educate the next generation of brilliant minds to challenge, explore, and advance ideas. Now, more than ever before, the world needs these ideas and passionate people to drive them. We are lucky to have some of our several homegrown Falling Walls alum joining us later today to hear about their experiences. This includes UQ PhD student and 2019 Young Innovator of the Year, Reese Pyrie. In 2019, I was lucky enough to travel to Berlin with Reese, as well as our former Vice Chancellor, to see him pitch a brilliant idea in just 3 minutes. He proposed a way to process waste glass into everyday products, which would potentially save thousands of millions of tons of glass from going into landfill every year. Alan Robinson will also be joining us today. Alan took out second place in last year's Falling Walls Australia event with his startup Clear Sky Genomics. The technology empowers patients to access and easily understand their own DNA and is the first doctor-patient centered approach to genomics. I hope that you all enjoy hearing from Reese and Alan about their incredible innovations in the session later today. Don't forget that today's event is also an opportunity to celebrate your work and to forge valuable connections with each other. It's through these kinds of connections that we can help to unleash new ideas that create a more healthy and sustainable future. We genuinely believe that you are the best resource that the world has for finding innovative solutions to some of the greatest challenges of our times. At the heart of this event is the question, which are the next walls to fall? I, for one, can't wait to hear about the research breakthroughs that you are, no doubt, all about to pitch. So thank you very much and all the best for the competition today. Thank you.
Thank you, Jessica. Now let me please introduce our amazing jury. They are joining us today to put our finalists through their paces. We have, as chairman of the jury, Professor Michael Rosemann, Honorary Consul of the Federal Republic of Germany and Director of the Center for Future Enterprises, Queensland University of Technology. We have Shannon Willoughby, Executive Director, Study Queensland. Professor Jessica Gallagher, Pro-Vice Chancellor of Global Engagement and Entrepreneurship at UQ. Professor Wen Yi, Pro-Vice Chancellor International, University of Southern Queensland. Leanne Kemp, Founder and CEO of Everledger. Vinod Krishnamoni, Director of Engineering, Oracle Labs. And Roger Tang, Chief Executive Officer and Co-Founder, Envirometrics.io. Thank you, Joey, for joining us today and giving the time to this initiative and to supporting the 10 researchers who will be pitching to you today, as well as making the wise decisions that they need to progress to the finale. So now let's see who will be presenting today. We have the Breaking the Wall of Waste to Energy, Mr. Arinze Zieke, the University of Queensland. Breaking the Wall of Parkinson's Disease, Mrs. Sarah Jose, the University of Queensland. Breaking the Wall of Vaccinating in the Homeless, Mr. Jeremy Hunt, the University of Queensland. Breaking the Wall of Suicide, Mr. Luke Bayliss, University of Southern Queensland. Breaking the Wall of Genetic Linkage, Mr. Apurva Ray, University of Queensland. Breaking the Wall of Lost 3D Opera during COVID-19, Mrs. Tara Rose, University of Queensland. Breaking the Wall of Prostate Cancer, Treatment and Resistance, Mr. Shamikara Linganage, University of Technology. Breaking the Wall of How We Feel Our Babies, Mr. Guillermo Tokman, University of Queensland. And finally, but not least, Breaking the Wall of Unsustainable Tourist Behavior, Mr. Yuhang Zhu, the University of Queensland. During the jury's deliberation at the end of the presentations, we have the opportunity to vote for the favorite presenter and topic for our People's Choice Award. So this applies to all of you in the audience. While the jury is in deliberation, we will also be joined by some previous Falling Walls Lab winners to ask us some questions and learn more about their experience in presenting Australia at Falling Walls. So today, our hashtag, and I think I see it on the screen, is #FallingWalls, and you can tag @UQ Ventures and @Falling Walls. And you see the RCD code, we all know these codes by now, right here on the screen. So, without further ado, let's begin the presentations. Each presenter will have 3 minutes to pitch their project. This will be followed by questions from a jury member to the presenter. So just as a reminder to our audience at home, keep track, do your own scoring, and you can participate in the People's Choice Award. So let's start with our first presenter. Please welcome our first presenter, Marianne Zieke from the University of Queensland, who will be presenting on Breaking the Wall of Waste to Energy.
Have you ever imagined a world where waste can be considered a resource? A world where all agricultural generated residues can be converted to valuable products? Yes, in my research world, all that you see as waste, I see as a resource. Similar to most countries of the world, Australia's berry sector produces tons of tasty fruits that contribute significantly to our economy. However, about 20 to 30% of these fruits are rejected annually due to quality standards. Most of these fruit rejects end up in landfills or are left open in the farms, contributing to environmental degradation, by proxy posing a threat to the health of you and I. The farmers are burdened with the challenges of finding the best possible way to manage not just these fruit rejects, but other residues such as the coconut coir coming from the fruit cultivation. My research focuses on finding the best possible approach to sustainably harness this so-called waste into resources. Anaerobic digestion is the key technology in my research. This basically involves feeding a group of microorganisms with these fruit rejects, which in the absence of oxygen are converted to energy in form of gases. However, it's not as simple, considering that the acid contained in these fruits could affect the balance within the microbial community. Imagine a football team of 11 players with different responsibilities, facing a tough opponent. For this team to win the match, the coach has to constantly tweak the formation so as to establish a balance amongst the players. I am the coach in this research. The microbial community are my players, while the fruits are the tough opponents. By tweaking the way of feeding the microorganisms, a balance can be established within the community, which will be more simple and cost-effective than current practices that either utilizes a cost-intensive and complex dual system that separates the microorganisms into two distinct communities, or by co-managing these fruits with certain and ideal ways such as the livestock manure that might not be readily available in certain localities. By utilizing the approach proposed in this current research, maximum energy can be recovered from the fruit rejects. A part of this energy can be used to power the anaerobic digester and return to the farm as a source of electricity and heat, while the other part can be used to power other systems for the management of the semi-solid effluents from the anaerobic digester and the coconut coir that would be difficult for the microorganisms to digest. The output from this other system will be nutrient-rich ashes that can be returned to the anaerobic digester as a supplement, or be returned to the farm as a source of fertilizer for the crops. It is my belief that the outcome of this research would be a win-win situation for all stakeholders involved. Myself as the researcher and the coach of these microorganisms, the farmers that will see the farm become energy and nutrient self-sufficient, the climate that will experience less release of greenhouse gases such as methane and carbon dioxide, and of course, the government that will see the waste management challenge of a key sector in high economy under control in a rather sustainable manner. Thank you.
Thank you, Arinze, for presenting on breaking the wall of waste to energy. Let's now welcome our jury member, Vinod Krishnamoni, to ask a question to Arinze for his comments.
Thank you, Arinze, for that brilliant pitch. So the question that I have is, in terms of the feasibility studies, what sort of feasibility studies have you done to prove the concept? And the follow-on question from there is, what kind of challenges do you see in terms of implementing this at scale?
Hello, thank you. So, so far, what we've done is lab scale, but it's really easy to really scale up from lab scale to farm scales. And this I have done actually with the energy analysis, where I've been able to actually establish that this is feasible in terms of energy viability. Then on the second question, sorry if you could tell me the second question again.
Yeah, the second question is what are the challenges in terms of implementing this at scale?
Okay.
So when you take it out from lab to the industry, right? And what challenges do you see there?
The challenges I foresee actually is on the downstream system. So the other system here mentioned in the presentation is a combustion system called smothering. So basically the smothering process has not really been used for this kind of waste, but it's an energy efficient combustion process. And it doesn't really require a constant energy source. That is why I actually identify these as a key component of what I'm doing. So the economic aspect is what could be a hindrance for mega farms here, but not for in terms of commercialization. If we consider mega farms and decentralized farms, it could present a little bit of challenge. However, it can be reimagined that you have maybe a central system where you have all these mega farms. For example, in Kabucha area of Queensland here, you have a lot of farms that produces berries that are into berry crop cultivation. So this, if we could actually cite a system in a central position where these waste can be pulled together and resources extensively valorized, gotten from this waste. Thank you.
Thank you.
Thank you, Arinzi Izieke and Vinwat, for this lively discussion. Now let's please welcome our second presenter, Sarah Uyeze from the University of Queensland, who will be presenting on breaking the wall of Parkinson's disease.
Imagine your life with tremor, stiffness, and pain, where simple daily activities like holding a glass of water, unlocking the doors with a key, or unzipping a bag feels like Herculean task. Imagine suffering from these conditions and knowing that you couldn't get better. A harrowing ordeal to think of, isn't it? The most recent Global Burden Disease Survey found that Parkinson's disease is now the fastest growing neurological disorder worldwide, with increased prevalence being linked to an aging population. The worldwide burden of Parkinson's disease has more than doubled over 26 years, from 2.6 million to 6.3 million by 2015. It is estimated that by 2040, it will rise to around 20 million people. This disease has reached pandemic proportions and exerts an enormous financial burden on healthcare system. Sadly, the biggest challenge is that we don't have any cure for the disease. The drugs that are currently available in market only temporarily manage the symptoms and do not stop the relentless progression of the disease. Consequently, there is an urgent need to develop new therapies and drugs that can stop the progression of this condition. In Parkinson's disease, as a person ages, there is gradual accumulation of protein aggregates in the brain, which persistently activate the immune cells in the brain. These activated immune cells trigger inflammation, thereby leading to nerve cell damage. This sets up a vicious cycle of chronic neuroinflammation. Metaphorically, I would like to call it as the brain on fire. Therefore, my approach is to halt this detrimental effect on the neurons and help the victims of Parkinson's disease. My next question was, how do I cool the brain down? In groundbreaking recent research by our group and other leading neuroscientists, we have identified that a key protein called NLRP3 inflammasome enhances neuroinflammation. Interestingly, in our further studies using mouse models of Parkinson's disease and in our laboratory experiments using immune cells, I have identified a key mediator protein in the NLRP3 pathway. Our results suggest that this finding could be a watershed in the treatment of Parkinson's disease. Thus, modulating this novel protein pathway could potentially stop neuroinflammation and help us break the wall of Parkinson's disease. Thank you.
Thank you, Sarah, for presenting on the breaking wall of Parkinson's disease. Let me now welcome our jury member, Dr. Jessica Gallagher, to ask the question to Sarah.
Great presentation, Sarah. Thank you so much. I wondered, you've indicated that, that this is currently in mice models. I wondered, what's the time frame in terms of when we might be able to see a drug of this kind come onto the market to start to assist people that are inflicted with Parkinson's?
So till now, we have done preliminary studies to see the target regulation in both in vivo and in vitro models. In both of the studies, we have seen exciting results, which has led us to do our further validation of target using genetically modified mice. We should have our genetically modified mice in a few months, and it would take a year to get some information from that. And once we get information from our in vivo knockout mice, then we can move forward. Then simultaneously, parallelly, we are also looking into the druggability of the target. And usually, as we know, that the drug usually takes 10 to 15 years to get into market with all the studies and stuff. So I think that would be a time frame for, if you ask me, the drug to come to market. Yeah.
And just in terms of what the competition looks like, are there a lot of other groups that are doing similar research, or is this quite sort of pioneer?
So definitely there is a lot of research going around, especially Australia being involved. So in my research, basically, I'm looking into the upstream regulators of inflammasome. And what I am doing is completely novel, and the protein that I have found is completely novel. So, but definitely with respect to inflammasome and the pathway, there is a lot of research going around. Yeah.
Probably needs all the help we can get. Thank you, Sarah.
Thank you.
Thank you, Sarah and Jessica, for the lively discussion and elaborations. Good. Now, please welcome our third presenter, Mr. Jeremy Hunt from the University of Queensland, who will present on breaking the wall of vaccinating the homeless.
Every year around April, I go to my GP, pay $20 and get my flu shot. I'm sure many of you do too. But for people experiencing homelessness, it's not that easy. They are disproportionately affected by the flu, more likely to catch the disease, and more likely to have worse outcomes if they do get infected. What's worse, often they cannot access the vaccine. The cost of the vaccine poses a major barrier. The federal government funds flu vaccines for high-risk groups under the National Immunisation Program. People over 65, people with chronic medical conditions such as asthma, and Aboriginal and Torres Strait Islander peoples. Bizarrely, people experiencing homelessness are not on that list. After discovering this as 2020 drew to a close, I set out to see if I could make a positive impact. I gathered some colleagues from across diverse fields, and together we established the Vaccine Project, an ACNC-registered health non-for-profit. We talked to several health clinics that specifically service homeless communities in Brisbane, confirming that this was a real problem that their patients faced, a problem that they were not able to solve for lack of funding. They told us that their patients were keen to get immunised, but they often missed out. In 2021, we raised money through community and grassroots fundraising to allow 200 people experiencing homelessness that otherwise would have remained unprotected to access a free flu vaccine through our partner clinics, Inclusive Health and Brisbane Youth Service. However, as we have come to realise, cost is only one barrier. Another is accessibility. People may not want to or be able to visit a health clinic. In 2022, we are establishing pop-up mobile outreach clinics to help visit people in hostels, boarding houses, and work alongside other existing services to vaccinate people in places that they feel most comfortable. We are expanding to support the homeless community in Toowoomba, as well as continuing our current relationships with health partners in Brisbane. We hope to support upwards of 500 people next year and help provide support to all 20,000 homeless in Queensland in the long term. Recognising the need for a sustainable funding model, the Vaccine Project is establishing a corporate flu vaccination arm, utilising a social enterprise model to diversify our revenue streams and magnify our positive impact. We are also in discussions with Queensland Health to see if there is a potential for us to work together and with other organisations to help deliver COVID-19 vaccines to those experiencing disadvantage in the coming months. I'm very proud to introduce you to the Vaccine Project, providing free flu vaccines to those who need them most. And we are excited to support people experiencing homelessness and help them protect themselves against the flu.
Thank you, Jeremy, for presenting on breaking the walls of vaccination the homeless. Now I would like to welcome our jury member, Shannon Willoughby, to ask a question to Jeremy.
Jeremy, I've got a question. How do you, you're planning on getting some funding through state governments and other governments, but how do you sort of scale this up given it presents such an opportunity with COVID at present?
Yeah, I think part of what we're trying to do, I know we're moving away from relying on grants and kind of the grassroots fundraising and trying to establish a sustainable fundraising kind of revenue stream. And part of that we think is going to be through the corporate flu vaccination service. So we're hoping to, alongside our charitable work, almost set up a social enterprise kind of model where on one hand, we're going to workplaces like a bank or an office block and setting up and doing the corporate vaccination there. And the profits from that section of the enterprise are then used to fundraise through and provide the services to people experiencing homelessness or other disadvantage. And so in that way, I think as we scale up on both ends, if they can scale together, then that will really have a great synergy that will allow the whole project to expand.
Good. Another question, we could ask Professor Ren Li to ask the second question.
Thank you, Hans, and very good and Jeremy presentation, and very timely as well. And is there any, you're looking for Australia, is there any advice or any ambition you can expand your project overseas? And like country, like have a large homelessness like United States?
Yeah, I think at this stage, I'm cognizant of the time here, I think at this stage, we're focusing on making sure this works in Australia and scaling it up across all states in Australia. But yeah, down the line, we definitely love to see across the world, especially in kind of countries like in the continent of Africa or Southeast Asia, as well as the United States. But yeah.
Good. So thank you, Jeremy. Thank you, Shannon and Professor Ren Li, for your questions and the discussions. Now, let's welcome our fourth presenter, Luke Bayliss from the University of Southern Queensland, who will be presenting on breaking the wall of suicide.
In every community, there are suicide wars. Wars of people who are in a suicidal crisis, with over 800,000 people taking their own life every year. 50 years of suicide research to identify who will take their own lives have left us with no better than random guessing. Not exactly helpful for breaking down the wall of suicide. So what's the difference between someone who thinks about suicide and someone who attempts suicide? People who attempt suicide do so because they have developed a capacity to act on suicidal thoughts. It's this capacity that facilitates the movement from suicidal thoughts to suicide attempt. But suicide capacity research is mostly focused on single risk factors. Focusing on one factor at a time means we don't get the whole picture. And subsequently, many of our suicide prevention strategies don't reflect the complexity of suicide, thus missing opportunities to break down walls of suicide. Continuing to research suicide capacity in this single factor manner makes it difficult to understand how capacity develops and what potentially affects it. It is difficult to develop suicide prevention strategies if we don't understand how and what moves someone from suicidal thoughts to attempt. This is what my PhD aims to do. Despite the want and the need to break down walls of suicide, we don't have evidence to design multifaceted suicide prevention strategies yet. First, we need to understand how capacity for suicide develops. My research is innovative because it is built upon suicide attempt survivor experiences, people who evidently have a developed capacity for suicide. It is their lived experiences that will help us better understand the movement from thoughts to attempt and provide insight into the development of capacity. But what about potential fluctuations of suicide capacity? We tend to think about suicide capacity as static and unmoving, much like a wall. But those with lived experience talk about their capacity varying. And this means that we need to identify contextual factors that affect changes in capacity. So, unlike any other research, my research utilises mobile technology for people with a lived experience of a suicide attempt to complete questions about their suicide capacity several times a day over multiple weeks. This will capture changes and contextual factors associated with these changes in real time. When we better understand how suicide capacity develops and what affects that capacity, then, and only then, can we develop truly multifaceted suicide prevention strategies that can save lives. Thank you.
Thank you, Luke, for presenting how you might break the wall aside. Please let me invite our jury member and jury chairman, Professor Michael Roseman, to ask the question for comment.
Thank you so much, Hans. Congratulations, Luke. This is a challenging and very important topic. Two questions. One, how do you ensure that those that you will interview have the reflective capacity, can give you the answers that you're seeking, so they're consciously aware of what you're looking for? And second, can you give us an example for contextual factor that you try to identify?
Yes, thanks for your question, Michael. In terms of reflective capacity, what we've taken is an inductive approach. So it's an unstructured, open-ended discussion, which is one question. Can you please tell me what led to your suicidal crisis? So then we start with the crisis at the end, and then we move back. And as people reflect, then we can take a narrative analytical approach to discover what contributed to the development of their capacity. And what's the second question, sorry?
An example for contextual factor that you'd like to identify.
At the moment, contextual factors, we don't actually have much data or knowledge. So the idea is that we'll conduct ecological momentary assessment. And through these assessments, we will have people complete surveys multiple times a day over a longitudinal design. And during that time, we'll be able to capture in a natural environment what's contributing to those increases or decreases of their capacity. And hopefully this will help us identify when people are at an acute risk for acting on suicidal thoughts. Then we can truly have multifaceted suicide prevention strategies to capture the movement and development of capacity, but also that acute risk stage, which is, highly valuable to preventing suicides.
Thank you so much, Luke, for those clarifications. This is very, very important work, and I wish you best of luck for the next steps.
Thank you, Michael.
Thank you, Luke, and thank you, Professor Michael Roseman, for going into depth in this important topic. Now, let's welcome our fifth presenter, Mr. Apubwa Ray from the University of Queensland, who will be presenting on breaking the wall of genetic linkage.
Welcome to my presentation on breaking the wall of genetic linkage. Purple maize in the world is not sweet at all, and sweet corn in the world is not purple. So what's the reason for this? The reason is a very tight genetic linkage that stops sweet corn from being purple. This genetic linkage means the purple color and non-sweet tastes are linked together, and sweetness is linked to non-purple characteristics. So to develop purple sweet corn, the first problem is there is an extremely tight genetic linkage between the dominant A1 anthocyanin producing gene and recessive SS2 SHANK2 gene. That means 1 in 1,000 plants can be purple sweet, but it's not straightforward. Otherwise, it would have been done before. The other problem is we need the expression of structural as well as regulatory genes. That means we need the A1 anthocyanin producing gene, and we need the SS2 sweetness producing gene. By control field experiments, out of a field of 6,000 plants, I have broken down 5 genetic linkages and developed the world's first purple sweet corn. Now we can produce billions of purple sweet corns. I have also completed biochemical, molecular, and genomics studies and found that the anthocyanin and sugar-related genes are expressed in my purple sweet corn lines. So what's the benefit of this research? The anthocyanin is produced as a product of purple color. So this purple color is responsible for preventing hypertension. That means you can get rid of high blood pressure. And another benefit of this purple color, that means anthocyanin, is that it prevents cancers. So what's the novelty of this research? The novelty of this research is that by breaking down tight genetic linkage between different characteristics, we can develop naturally nutritious products. Those are not genetically modified organisms. So we can get the nutritious plant products out of this kind of controlled field experiments. And this purple sweet corn is now ready for startups and industrialization. Thanks for hearing. Any questions, please?
Thank you, Apurva, for presenting on breaking the walls of genetic linkage. Now let me welcome jury member Liane Kemp to ask the question to Apurva.
Hi, everyone. Funnily enough, I had corn for dinner last night, so I was very excited to be the person to present a question to you today. Have you had any large regulation boundaries that you've had to alleviate in being able to see commercialization of this research?
I think not that much problem for commercialization, because this is a project of hot innovation of my supervisor, Associate Professor Dr. Tim O'Hare of University of Queensland, of hot innovation project, and also the University of Queensland has funded my scholarship. So for industrialization or starting up something, I think the university and the project have some supports, because we need to actually take our invention to the consumer's level. And also my supervisor has completed a consumer evaluation, and it's fascinating for this purple sweet corn industry.
Good.
Queensland is very strong. Queensland is very strong with synthetic biology. So definitely reach out to the chief scientist, because he would be very interested in this work.
Yeah, should be.
Okay. Thank you. Thank you, Apurva, for presenting on breaking the walls of genetic linkage, and good luck with the project. Now we would like to present the next presenter, who is Tana Rose from the University of Queensland, and she will be presenting on breaking the wall of lost 3D opera during COVID-19.
Hello, my name is Tana Rose. Today I shall be talking about breaking the wall of lost 3D opera during COVID-19. The problem that I am trying to solve is how to create opera experiences which can be viewed during the pandemic. The solution is to create 3D opera utilizing extended reality technologies. What are extended reality technologies, you may ask? Extended reality technologies include virtual reality and augmented reality. I'm a virtual reality developer and augmented reality developer, creating a new opera called Artemis. This opera is employing singers and pianists from the UQ School of Music and Greater Brisbane. So I have created new models for prototyping new opera creations. These models can be utilized by independent extended reality opera creators. If you would like to see some of my prototypes, you can Google Tana Rose Music, and you can find arias from my opera Artemis on YouTube. These arias can be viewed with an Oculus Quest headset or a Google Cardboard VR headset. There is also an augmented reality film of Artemis which can be viewed on your computer. I paint the virtual reality art and augmented reality art, which I utilize in the production Artemis, with tilt brush. This software allows me to paint in real time in the 3D environment.
Thank you, Tana Rose, to tell us about how you will break the wall of lost 3D opera during our pandemic COVID-19. Let me welcome Dr. Jessica Gallagher to ask the questions to Tana.
Thanks, Tana. It's a really interesting concept. I wonder if you could tell us a little bit about the sort of motivation in terms of this particular research. What sort of drove you to look at opera in particular?
Absolutely. Thank you for your question, Jessica. Basically, I am a music technologist and a composer, and the future of music dissemination is really online, especially now with the pandemic. So I started my PhD prior to the pandemic, and my supervisor suggested utilizing VR and AR to capture that market and the future of technological opera.
Mm-hmm. I might pass it over to another panelist for another question.
Thank you.
Good. So,
thank you.
Vinod?
Yeah, thank you, Hans. And thanks, Tana. That was a very interesting presentation. So in terms of the equipment that you used, right? I think you mentioned Oculus and Google Cardboard, and I think they come with their own tools that will allow you to create the models. So did you have any unique computing problems that you had to solve during the course of coming up with these new models? I mean, that the tools couldn't do? I mean, did you find anything like that? And what approach did you take to solve that?
Sure, thank you for your question, Bernard. Basically, I use with Unity 3D software and utilizing pre-built prototypes that you can buy from the Unity store. Some of those had compiler errors. So in the end, I decided I will switch to using tilt brush and creating my own virtual environments from scratch by painting them rather than buying preset models and implementing them. However, I did utilize some free preset models from Unity, and those worked well, and I've also created a working model in that way. So I have two prototyping methodologies that I'm employing.
Right. Okay. And the toothbrush, but that was again off-the-shelf software. Was it open source or commercial?
Yes, so that's off the shelf. So that's commercial, and you buy that in your virtual reality Oculus Quest headset. That's correct.
Okay. All right. Thank you, and good luck. Thank you.
Thank you, Tana. Thank you, Jessica and Vinod, for exploring this project further. Now, please let me welcome our seventh presenter, Mr. Shamikara Linanaji from the Queensland University of Technology, who will be presenting on breaking the wall of prostate cancer treatment resistance.
You can't hide yourself after committing a crime. However, cancer has been always smarter compared to us to hide themselves from all our treatment regimens. This has been true for prostate cancer as well, the most common cancer among Australian men. 1 in every 6 Australian men has the potential to be diagnosed with prostate cancer by the age of 85. But being diagnosed with prostate cancer is not a death warrant at all. Until the cancer remains only in your prostate, it can be treated with surgery or chemotherapy. But with the time, cancer cells start adapting to the new environment and escape from the treatment to other body parts, such as bones, lungs, and liver. And it is almost impossible to treat these advanced prostate cancers, and the survival rate falls less than 30%. In year 2021 alone, more than 3,000 deaths are estimated for men having advanced prostate cancers who has undergone extreme level of chemotherapy. For this reason, researchers are always searching for new treatments with better disease control and longer survival rates. finding answer to this question. We took a deep dive into cancer cells where nobody has ever looked on. For the first time in prostate cancer research, we discovered a group of microproteins hiding inside cancer cells that might provide an answer to break the walls of treatment resistance. Microproteins have been continuously ignored by researchers because of their smaller size. And the surprising fact is, these microproteins are produced by non-coding genes, which was previously believed as genes that do not make any type of protein. The key breakthrough of this research, it's not just the discovery of microprotein, it's all about their biological role. So we used a viral-based gene delivery method to reintroduce these genes into cancer cells and animal models to produce sufficient amount of microprotein copies. And with that, we have seen a greater reduction in cancer growth and their potential to invade other tissues, suggesting the cancer-inhibiting role of microproteins. These evidences has given us with the foundation to use these microproteins in gene therapy for advanced prostate cancer patients. Due to their high level of activity, use of microproteins in gene therapy is highly reproducible, and new modifications can be introduced to improve stability, safety, and their biocompatibility. Not only that, if we combine gene therapy with existing cancer treatments, we can increase patient survival by breaking the walls of cancer treatment resistance. We believe that discovery of microproteins will not just shape up the legacy of cancer therapy, it will open up new avenues in modern medical sciences. Nevertheless, these microproteins are still the lilliputs in cancer research, but never underestimate little things, because little things can make big things happen. Thank you.
Thank you, Shamikawa, for presenting on breaking the wall of prostate cancer treatment resistance. Let's now welcome jury member Professor Michael Roseman to ask the question of Shamikawa.
This was an outstanding presentation, a big topic, and I like your presentation skills. For us as non-experts, can you tell us why have microproteins not been explored earlier? Because I assume this is a highly competitive area of research.
That's an interesting question, actually, Michael. So the previous, microproteins was there in the cancer biology from the beginning. So what was the question was like, people hasn't been looking on these microproteins because the methods that we were using, like high mass spectroscopy methods, were not sensitive enough to identify these microproteins. So we have developed a highly sensitive SWAT-MS-based mass spectroscopy method to identify these genes using both genomic and proteomic expression data. And the surprising fact, as I mentioned, these genes are coming out from non-coding genes. So there are almost 50,000 non-coding genes in the human genome, and they have been probably wrongly annotated. Most of them are wrongly annotated. So we believe, we have developed a novel database, including this proteogenomic approach, to identify these possible microprotein inside cancer cell. And another exciting fact is these microproteins are not largely expressed. So people believe that the proteins that are not highly expressed are not valuable enough. But we have already proved, and there are already previous studies showing that microproteins are very less expressed, but at the same time they involved with highly important cellular mechanisms.
We've got 25 seconds left. What is the biggest risk to your successful research?
The most risk for my research is associated with the safety precaution with the gene therapy, which makes these clinical trials more expensive and cumbersome. But with acquiring the past knowledge and acquiring 40 years of knowledge about the viral vectors, we have developed new viral vectors, which is less toxic, and new modification that become these viral vectors are more specific to the cancer treatment, and while balancing the results of what we have seen in the animal models. So that is the major challenge, but we have already overcome that.
Thank you, Shankar.
Thank you, Shamikawa, and thank you, Professor Michael Roseman, for exploring this in depth. Now let me welcome our 8th presenter, Mr. Guillermo Tocman from the University of Queensland, who will be presenting on breaking the wall of how we feed our babies.
Falling walls of baby nutrition. Parents are worried about their baby's well-being. They know that human milk has all the nutrients that their baby needs, and parents who cannot breastfeed ask themselves if their babies will grow healthy and strong. Let's see what is happening today with the current infant formulas. The majority of infant formulas use cow milk. And if you see, cow milk doesn't have essential antibodies that human milk has. So if babies are not calves, why we feed them like one? We believe that we have a solution. We want to create the first, second best choice to feed your children, narrowing the gap between breast milk and formula. We are going to produce a baby formula closer to the human milk, adding at first stage the main antibodies that baby needs, focusing on the strength of the baby's immune system. We have a patented recipe and the price will be in line with the current baby formula market. No competitor is offering a complete alternative. Some are offering product based on goat or soy milk, and others are offering a basic ingredient formulation. We ran a customer validation that includes 106 Australian parents, and we could validate that more than 80% will or would probably buy our product. As we know that our plans are bold, we propose a 3-horizon plan. First, we will create brand awareness and put our R&D efforts to synthesize the antibodies. Second, we are working with the proteins and carbohydrates to stop using any cow milk in our formulation. And finally, with the fibers and fat synthesis, we will produce the closest human milk produced ever. We have an experienced team. I'm a food engineer with more than 10 years of working experience at the baby formula category. And in the team also we have a doctor in biotechnology with more than 30 years of experience launching medical supplements, and a marketing expert with more than 8 years working experience in top multinationals. We are committed to create the best product possible. And we know that we have the capabilities to do so. We want to help parents in such a meaningful part of their lives. So let's nourish our children together. Thank you.
Thank you, Guillermo, for presenting on breaking the wall of how we feed our babies. Now let's welcome our jury member, Liane Kemp, to ask questions of Guillermo.
The baby formula market's a staggering $100 billion market. There are significantly large companies in the world that are dedicated to making these formulas. Why do you believe that your patented work is a breakthrough technology per se that hasn't already been discovered or implemented quietly by these large companies that dominate the space?
Yeah, sure, thanks. But the idea is to have a more complete product. Actually, as I told in the presentation, nobody's offering a kind of complete product in the way that there are a lot of antibodies that human milk has and cow milk not. And actually the proteins, the fibers are not the same. So at this point, these big companies did a good job, like trying to replace in one way when parents cannot breastfeed. But our idea is go beyond that, like have a closer and have almost the same component that human milk has. And for sure, it's a lot of R&D efforts on that. And I worked in one of these big companies, and I think that the work so far was good. But I think that now with all the biotechnological revolution, we can go beyond. We can go ahead with a better product.
Is your ambition to commercialize the patent, or is it purely to license the patent?
Now, the idea is to have a product, and actually with the following years a product range. And the good thing about Australia is that we are located strategically with four countries in the region, and the US has the 50% of the volume of the infant formula worldwide. So I think that there is a good opportunity to be here too.
Good. Thank you, Guillermo, and thank you, Leonie Kemp, for exploring this topic further and the ways to bring it to market. Now, please welcome our ninth presenter, Johan Oscar Zhu from the University of Queensland, who will be presenting on breaking the wall of unsustainable tourist behavior.
Hi everyone, I'm Oscar from the University of Queensland. Today I'm here to break the wall of unsustainable tourist behavior. We all like traveling because it gives us a great opportunity to rest, relax, and recover by spending some leisure time in a completely different environment. It is not only the environment that changed, our behavior actually changed as well. We view vacation as a very special time. We give ourselves permission to behave less responsibly than we do at home. We abandon many of our environmentally sustainable habits. We enjoy extra long hot showers. We run the aircon all day with the window open. And we pile up our plate with food in a buffet and leave most of it behind uneaten. Even people with a super poor environmental value do not carry over their environmentally friendly behavior to the vacation context. So it's not surprising that tourism is among the most polluting industry in the world and contributing 8% to global greenhouse emissions. So how am I going to break this solid wall? I propose to fight fire with fire. So the reason we do not behave in environmentally sustainable way on vacation is because we want to enjoy ourselves. So why not we make the environmentally sustainable behavior enjoyable, or even more enjoyable than the unsustainable alternative? It is possible. One study used a stamp collection game for families. So every day in the dinner buffet, when families ate up all food they took, they got a stamp. So if they collect a stamp from each day of their vacation, they receive a prize at the end. Kids love it, families love it, and food waste was significantly reduced. Breaking this wall will have a huge impact on multiple stakeholders. Tourists will have more fun on their vacation. Tourism operators can reduce their operating cost, like food cost in the example with the stamp collection game. And the biggest winner, of course, is the environment. Making tourists behave in a more sustainable way is very difficult, no doubt about that, but it's not mission impossible. We can fight fire with fire. We can use the very driver of unsustainable behavior and use it to trigger sustainable behavior. The focus on maximizing enjoyment. Thank you very much, and any question is welcome.
Thank you, Johan or Oscar, for such a lively presentation on breaking the wall of unsustainable tourist behavior. Let's now welcome our jury member, Roger Tang, to ask a question of Oscar.
Thank you very much for that. I really enjoyed that presentation. But I just want to ask, outside of the hotel and buffet situation, how do you plan to expand this to encourage more sustainable behaviors? Interesting.
I'm sorry, you mean the outside the hotel context? So yeah. Actually, it actually should be more generalized to the hedonic context. For example, it's any enjoyment focused context. The hotel is just an example. For example, the enjoyment focused context could be grab a coffee, you know, like where we like between the work, like anything make us feel like we want to relax or enjoyable. This is kind of like enjoyment focused context. So the hotel is a rough example because it's where the most problem, you know, gathering together in tourism industry, and it's more easier to kind of tangible the effect of the intervention. That's why I use the hotel as an example.
Okay, and what do you see as the I guess the major problem to tackle to reduce, well, at least to make tourism more sustainable?
Yes. Sorry?
What would you say is the major problem to tackle to make tourism more sustainable?
Well, definitely because people think, like you in your mind, you think perform in a sustainable way somehow harms your enjoyment. For example, when you in a hotel, you want to enjoy yourself by the aircon, the shower. So people in the mind, they kind of like connect the environmental behavior and as unpleasant. So I think this is the biggest challenge. Like people in the holidays, they just want to relax. So they will refuse to do anything like will undermine their enjoyment unless you can, like my intervention propose, attach the enjoyment to the environmental friendly behavior. So kind of like encourage them to engage.
Okay, thank you very much.
Thank you.
Thank you, Oscar, and thank you, Roger Tang, for the exploration of this important topic. And I think we should now thank all of the presenters for, first of all, having such great ideas, and secondly, presenting them so clearly. So a choice has to be made, and the jury will now deliberate for about 20 minutes who will make it through to the national finale in September in Canberra at the Academy of Science. So the jury goes into their room. We should applaud or, have an invisible thumbs up for all of the presenters. And for all of you out there, please remember that you can look at your scores and you make your choice for the People's Choice Award. We'll see you soon, and I will have now a chat with some previous winners of Falling Walls Lab.
It's super important to be able to showcase talent like Reese, that it clearly is an exemplar for the types of innovators and creators and entrepreneurs and scientists that we have here in Queensland.
We've developed a process where we take the non-recyclable fraction of glass, which is the small pieces that can't be sorted back into the right colour, and we dissolve that in a chemical which most people know as drain cleaner, and we make another chemical which is called sodium silicate, and with that we can make things like tyres, detergents, silica gel and even toothpaste. I feel really fortunate to have done my PhD in Queensland. It's easy to access top quality researchers across a number of areas. So for me, I started in agriculture, went across to engineering, and I've ended up somewhere in the middle. Queensland
is a place where you can start here and go anywhere.
Good. So I hope I can very soon welcome two of the previous winners. Reese.
Hi Hans, how are you?
Can I see you somewhere, or are we jointly on the screen? Ah, lovely. Good to see you again, Reese. How are you?
Oh, not too bad. Good to see you as well.
Alan, yep, it has been a while. We traveled together in 2019 to Berlin. So can you tell us a little bit about your experience, what it was like to be in Berlin at the international version of Falling Walls?
Sure. It seems like a bit of a lifetime ago now. It was all pre-corona. And it was actually my second university-related trip to Europe that year, which seems a bit indulgent at the moment. But yeah, it was incredible in terms of the event itself. It's a long day with the pitch, so you kind of have a day before to meet everyone and socialise, and then when the actual pitch is going on, just getting through 100 pitches of 3.5 minutes each takes the whole day. And I was, I think, 97th or something like that, definitely into the 90s. So it was at the end of a long day, and then hopped up and gave the pitch, and thought it went kind of okay, but yeah, honestly wasn't sure where it would have landed, because it's cliche to say there's lots of kind of really good pitches, but there are, and I kind of thought, oh, maybe I should be somewhere in the top 20, but apart from that, I don't really know how it goes. And then, yeah, as the announcements rolled around, obviously I was announced as the winner, and so kind of went up and took the award. And then later that night, there was a really special event on at the Natural History Museum with a flash opera crowd that turned up and started singing. And yeah, it all sounds like a lot, especially I'm living in Sydney now, so I've been in lockdown for the last 6 weeks. So yeah, I'll probably stop talking there, Hans, and you can ask for any more details, or Alan.
Yep. No, I think this was a very, the good old days before the pandemic, when we were all traveling. Large crowds were allowed, and large crowds and meeting lots of people in a short time was one of the highlights, obviously, when we all get together. So can I have Alan Robertson on the screen?
Hello.
Hello, Alan. How are you?
Very well, thank you. How are you, Hans?
Good. Now, we never met in person, I think.
Oh, we haven't, and I'm very jealous because of it.
Yeah, so the problem is that for 2 years in a row now, we have Falling Walls online, but it hasn't diminished the ideas, it hasn't diminished the enthusiasm. So tell us a little bit about your experience when you went from Queensland to Australia, and then the bigger audiences.
Yeah, so I was very, very fortunate. So I snuck into the Australian final, so I came third. I then was very fortunate to come second in the Australian finals after Queensland. And it was a completely different experience just because of COVID. So I think with the Queensland one, we had to record videos. Then with the national level one, they zoomed in, and they tried to make it as live as possible. So they just recorded everything, and you just had one chance to get it right. Then when they did the international ones, and that was the where we could just step back. We had to answer, I think, 7 questions, but we could pre-record the answers, so we could do it as many times as we wanted until we got it right. And I still think I looked like a used car salesman in 90% of my takes, but it was a great experience overall. So it really allowed me to, I guess, focus on telling one version of my story for a specific audience. So yeah, a great opportunity, despite the fact we didn't get to go over to Germany.
So let me explore that a little bit further, because it might not be known to the people on the viewing us here and the presenters. So we are working with the format of the 3-minute pitch, which then Reese, you described you were giving that live. And I also recall that we made you give that pitch several times, right?
Oh, look, every time we did it, it got better. So
yeah, that's right. With Reese, there was an occasion at a pub 2 days before with the ambassador, the Australian ambassador.
That was probably the best.
That's when you really relaxed, right? Can you describe that moment when you pulled out the bottle?
Oh yeah. So I was fortunate that obviously with glass, you've got a ready-made prop. So I was kind of giving the pitch, and then obviously you had an empty beer glass in one hand, of being able to point at it. I do that comment you made, Alan, about sounding like a used car salesman by the time you fine-tune your pitch. And this might be one of the questions later, Hans, but I think that's something that I really focused on was because there's so many TED Talks, there's a very standard format of how people tend to present things. And I tried to make it a little bit more offbeat to make it different. And that was probably, I don't know, just if you're formulating your pitch, I think there's kind of those standard elements. You want to tell a story, you want to add human elements to it. But if you can have something a little bit different that's authentic and offbeat, and that captures people's attention as well.
Oh, absolutely. And I guess for me, that comment was me because I realized watching all the feedback or watching all the video back that I stand with my hands on my hips with one hand out. I just wanted to like slap the boot of a car and said, oh, this baby can hold so many genomes in it. I only had one owner. But yeah, it's every time you do it and every time it's filmed differently, it's a great way to refine the way that you present and that you see yourself presenting, because watching yourself back is very different from actually watching yourself giving it, if that makes sense.
Yeah, I mean, Zoom is a bad one, isn't it? You can see yourself all the time on Zoom, so I find that pretty uncomfortable anyhow. Rather be hiding. So it's been 2 years now since, or over 2 years since the Berlin one. Reese, what's the sort of biggest impact Falling Walls had on you or your work?
Yeah, so I guess there's the initial impact. It generated a lot of publicity for the research and the technology. So the commercialization is still progressing through UniQuest, and hopefully we'll have a bit more to say about that shortly. In terms of me, I'm working for New South Wales government now, and it definitely made my job application stand out. And in terms of one of the most common interview questions you tend to get in an interview is kind of, can you tell us about a time when you've had to communicate complex concepts to a standard audience? And it's kind of, oh, thanks for stepping into my hitting zone. So yeah, probably the biggest direct personal benefit for me at the moment has been it's definitely made me more employable, and then as well as kind of increasing your soft skills and network and all of those things.
Yep. And how about you, Alan?
Oh, I guess it's much of the same. So I guess the thing that really stood out was, I guess, how much it really meant to a lot of my collaborators and co-workers. So I'm like, oh, it's nice. We've got this thing and add a line to my CV. But I'm part of the Digital Health CRC, and they were just so impressed and so enthusiastic, and like they put out this media release. It was like very, very fortunate. So they really, really saw the value in this, and it was also nice to be officially labelled as an emerging talent of science by the Australian Academy of Sciences, which is something I like to bring out when someone's talking smack about me on Twitter. No, that's a joke. But, yeah, it was a really overall it was like a great experience just to kind of I guess highlight to people, I guess, the impact of what we're doing and really have independent validation of the importance of the work that we're kind of focusing on.
So let's talk a little bit about the journey in just in a brief time. How did it all start? I mean, how did you find the research project, Reese?
So yeah, in terms of my research project, I'm very much an engineer by accident. My PhD originally started looking at nutrient uptake by plants and soil as a media, and then I became interested in silica as a kind of addition to fertilizers to make fertilizers more efficient, and then looked for cheaper sources of more available fertilizer. And one day my supervisor suggested I could look at glass as a source of readily available silica. And I thought that was a stupid idea, so I laughed at him and went and tried to do other things until my confirmation deadline rolled around and I had no other good ideas. So I started then playing with the glass and kind of saw some interesting other results and refined from there. So yeah, it definitely wasn't a linear journey.
Like many are, I think, and you were always looking for the next possible big question to resolve. So was there a sort of aha moment, something that you remember where you said, yep, that was a leapfrog in thinking or experimentation?
I think the biggest aha for me was when we had produced something that was kind of approaching a commercial grade product, and then we did the rough kind of cost balance model, and there was a significant savings compared to what the conventional original product was. And that was the moment we thought, oh, hang on, if we're kind of greener and cheaper, that has a real commercial impact and, you know, might be able to be taken up by people. That was probably the biggest, oh, this could be really significant moment.
Alan, do you have a similar convoluted story, or?
It's convoluted, but just because it's the way that I tell it. So I'm from Queensland, and I've been at UQ for a very long time. And UQ was one of the leaders in genomics. So we had early access to the technology that made things like, you know, discovering the genomic aspect of microproteins possible. We had it way earlier than everyone else in the world. And I was very fortunate to kind of fall into that back in 2007. But the longer that we kind of spent in this space, the more we started telling people that how this could improve human health, but the less it really did. Like we'd go out there and we publish in Nature and we tell people about how our work was going to improve healthcare for everyone. But when the people we cared about got sick, they were still getting the same treatments and the same analysis techniques and the same diagnostic procedures that were being offered to them 20 years ago. And the longer I spent in this space, the harder it became to kind of reconcile that. And I kind of realized we could have a good whinge about it, or we could actually do something about it. And that's kind of what led me to go really look at the pipeline of genetics and, you know, kind of where the innovations are and what needs to be innovated in order to really facilitate the clinical adoption of this technology. And we kind of realized that in that pipeline, we'd really left the last end of it really untouched. So we spent a long time focusing on trying to support clinicians using genetics and clinicians communicating those results to patients and their families. And that's kind of what led us to Clear Sky.
So at some stage, you must have decided a startup company or some sort of commercial approach is the way forward. So tell us a little bit more about the role, how you see the link between the commercial world, which can have various forms, and what you want to achieve.
Look, to me, I'm not one of these people that was born with a briefcase in hand. I'm not, let's say, an entrepreneur by nature. I'm from Umundi, which is a small hippie town. So the idea of doing something commercial for them is like sacrilegious. But for me, it was a realization that all of the big advances we've had in medicine really relied on a commercial partner. And we could either say, oh, that's terrible, or we could say innovation is an avenue for commercialization is an avenue for translation. And if we're in control, we can be the ones making decisions to make sure that they're not negatively impacting people. So for me, it was really just using the processes to achieve my goals, which is helping more people understand the information in their DNA.
So you know how to make a pitch commercially, but you're still pretty relaxed in terms of the, you know, the way the companies operate.
Also, we're very focused, I guess, making sure the company operates, but in a very in a way that is consistent with our ethics. You know, and we're still making sure that we can provide like a viable business opportunity. You know, that's, you know, the way that things need to work in the business world. You know, they need to be able to return a profit, but we're also making sure that it's actually supporting, you know, human health. It's going out there to actually make the world a better place rather than, you know, making me fat stacks of cash.
How does that match your observations, Reese?
Yeah, I wouldn't say dissimilar. I would probably, for me, I think business for a large part is where science impacts people's lives. It's kind of, we can do all the interesting science in the world, but until that's turned into a product or a service that people can consume, it doesn't really impact their lives. And so, yeah, I think if you're kind of out there to change the world, you need an idea that's commercially promising as well, which is people aren't going to keep funding things in perpetuity if they're not seeing a return, or at least self-sustaining. So yeah, I think there's antagonism kind of there and has been in the past, where companies have done the wrong thing. But I mean, we look at current COVID situation. Who would have thought that we have nearly a dozen vaccine candidates, 12, 18 months, kind of using technology that had never been used before. It's absolutely incredible and only possible with the commercial incentives that have been provided to those companies. So, yeah.
Yeah, I think that's a very important observation. The moment we really depend on science more than ever to fix this particular problem. And so far, the companies are playing a very constructive role. I mean, you know, I could imagine in 2 or 3 years' time, the big profits will be made when we have booster shots for years on end and all of that. But, you know, that's as you said. Now, I've seen over the years that it takes many different type of people to make this commercially successful. You know, the creator, the shy person, the person out there doing the pitch, but then the people who runs the company, the CEOs, they're all very sort of different personalities. And you obviously found a niche in that. I mean, how is that collaboration going with people who actually have probably not even a science background, but think about the same problem in a very different way? Have you got examples for that, Alan?
Oh, absolutely. So I've been very fortunate enough to be supported by a fantastic venture group here in Brisbane who provide me with not only business support, but also access to their developers and designers, and collaborating with the developers to actually build a medical grade solution up to the standards required by the TGA, as well as a designer that takes all my complex thoughts and transforms them into something that can be understood at a glance, is a very collaborative, very exciting, at some stage very frustrating process. But it's really where being able to articulate your problem and the significance of your problem, which is what this competition is all really about, is, I think, the secret sauce to actually working together to do something with these big groups of people with a diverse range of skill sets. So making sure they all understand what they're working towards and why it's significant is just such an important part of any real high functioning group.
Good. Look, there's some real news coming in that the jury has actually made the decision. And so we'll be switching back to announce who's the winner of this year's Falling Walls Lab. It was a great pleasure, Alan and Reese, to chat to you. And I hope out in the audience gave you some insights how science is developing and how our winners are getting closer and closer to their goal. So good luck for the future.
Thank you very much.
Thanks, Hans. Good luck, everyone. And hi to the familiar faces on the call.
Good. Good. And I think we'll now switch back to the room with everybody in. The switching and forwards and backwards in the Zoom Live. And I've got the word that the jury has finished the deliberation. So let's have a look again before we announce the winner at the 9 presenters and their topics. So we had Breaking the Wall of Waste to Energy by Mr. Arinze Ezieke from the University of Queensland. Breaking the Wall of Parkinson's Disease by Mrs. Sarah Jose, the University of Queensland. Breaking the Wall of Vaccinating the Homeless, Mr. Jeremy Hunt, University of Queensland. Breaking the Wall of Suicide, Mr. Luke Bayliss, University of Southern Queensland. Breaking the Wall of Genetic Linkage, Mr. Apurva Ray, University of Queensland. Breaking the Wall of Lost 3D Opera during COVID-19, Mrs. Tara Rose, University of Queensland. Breaking the Wall of Prostate Cancer Treatment Resistance, Mr. Shamikara Naji, Queensland University of Technology. Breaking the Wall of How We Feed Our Babies, Mr. Gulnon Tokman, University of Queensland. And last but not least, Breaking the Wall of Sustainable Tourist Behavior, Mr. Yuan Oscar Zhu, the University of Queensland. Now, thank you to our fantastic jury who made themselves available. Chairman of the jury, Michael Roseman, Honorary Consul of the Federal Republic of Germany, Queensland University of Technology. Professor Jenica Gallagher, Pro-Vice Chancellor of Global Engagement and Entrepreneurship from the University of Queensland. Shannon Willoughby, Executive Director, Study Queensland. Professor Ren Yi, Pro-Vice Chancellor of International, University of Southern Queensland. Leanne Kemp, Founder and Chief Executive Officer, Everledger. Vinod Krishnamurthy, Director of Engineering, Oracle Labs. And Roger Tang, Chief Executive Officer and Co-Founder of Envirometrics.io. So I've been informed that the jury has been able to find 3 winners, and all of them would move on to the national final in Canberra. So please welcome jury chair Professor Michael Roseman to join us and announce the winners.
Thank you so much, Hans, and from all of us, thank you again for fantastic facilitation. From all of us, the judges, to all of you who presented, you are all heroes. Our task was just to find the very few superheroes. You are great scientists and great science communicators, and we all appreciated how you were able to combine evidence and confidence. It was very difficult, but finally, I think we agreed at a compelling consensus in terms of the top 3. And what I will do is I now list out the winners of the competition, starting with number 3. So the third highest ranked presenter studied and explores incredibly crowded area, and we like to congratulate Arinze Izike, who tries to break the walls when it comes to waste to energy. From all of us, congratulations, well done.
Jake, some short comments about winning. How do you feel?
I'm short of words. I thank you all, and I'm really excited that I will be going all the way to Canberra. I hope to achieve greater things. Thank you.
Congratulations again.
Thank you.
That brings us to, I think, the silver medal. The silver medal goes to a candidate who played with fire. Again, like waste to energy, the topic is a very crowded topic, and it's hard to stand out in crowded areas. We appreciated how compelling the context was set and how convincing the research questions are explored, and seeing first evidence of success made this a very, very compelling showcase. Putting the brain on fire is not an easy task. So from all of us, Sarah Joes, congratulations and good luck with your attempt to break the wall when it comes to Parkinson disease. Congratulations.
Congratulations, Sarah.
Thank you very much. Yeah, I feel very happy. I thank God. I thank everyone for giving me this opportunity. And I thank my professor and everyone in my team who helped me. Thank you very much.
Wow, and we are looking forward, Sarah, to seeing you in Canberra along with the other 2.
Thank you.
Well, now we come obviously to the gold medal. It is an incredible privilege to announce the final winner of the Falling Walls Labs 2021 here in Queensland. And before I do this, I'd like again to thank everybody involved, in particular the University of Queensland, for demonstrating your flexibility, Jessica, Megan, and the entire team, and also, of course, Shannon, and your tremendous support from Study Queensland. Ladies and gentlemen, so the final winner, again, is an area that is of tremendous importance if solved well. It's a crowded area, and crowded area bring the best out of researchers. What we liked here in particular is that sometimes small things matter, and as the presenter highlighted, from small things, big things grow. And it's true with your research as well. And we hope that some of the research presented here grows so big that it will be shown in Berlin in November at the final Global Falling Walls Labs. Ladies and gentlemen, it gives me tremendous pleasure to announce the winner of the Falling Walls Lab 2021 here in Queensland. And the winner is Shamikara Liyangi and his fantastic work on breaking down the wall when it comes to prostate cancer treatment resistance. From all of us, congratulations, Shamikara, and good luck in Canberra.
Yes, thank you so much. I'm so embarrassed. And I would like to thank the organizers and especially my supervisors in Queensland University of Technology and my lab mates for always helping me to achieve these goals, and my family back in there in Sri Lanka, and my mom, and everybody who supported wholeheartedly. And big thank for the organizers and all the winners, and everybody. I believe everybody did really well, and I hope everybody will get a chance in next time and how to present well in the Canberra competition as well. Thank you so much.
Congratulations again. Well done.
Thank you, Michael, and the whole jury for making such a wise choice. And we're really looking forward to have all three of you presenting again in Canberra for the national finale. And it's basically a clean slate. Yes, you have the gold medal from Queensland. There will be 10 or 11 or 12 now competing from across Australia. And you have another go at it and convince us all about the importance of your really fantastic research. So let me please announce the People's Choice. And the winner for today is Shamikara Liyangi. Again, so there was identity between the jury and the People's Choice. Over 50% of the votes went to you. But, you know, the next level of competition will challenge you again. So congratulations to winning both the jury and the People's Award.
Thank you so much.
So before we close, I would like to introduce and welcome Shannon Willoughby, Executive Director of Study Queensland. And they are a very valued partner of Falling Walls Lab. I'd like to ask Shannon for a few words. And how was your experience as a judge, if you could mention that at the end, please?
Thanks, Hans. Look, on behalf of the Queensland Government and Study Queensland, I just want to say a big shout out to all of the presenters today. It's always amazing to see the depth and breadth of talent that we have in Queensland and some of the amazing research that, you know, as a judge, sometimes it's hard to fathom how much work goes into a lot of that research, and then you have to articulate it back in 3 minutes time. So a big shout out and congratulations to all of you. It's a great learning curve. It's a great opportunity to be able to present in the Falling Walls Lab Queensland. This is the third year that we've had it here in Queensland, and Study Queensland and all of our university partners brought it to life with Hans and Dr. Roseman 3 years ago when we had our only face-to-face one. We've had to obviously pivot and move to virtual. So yeah, big shout out to you all. Congratulations, Shamikara and all the other winners. Well done. I look forward to seeing you, how your training and the opportunities that are presented before you go down virtually to Canberra. I probably just make a couple of reflections. 1990 was a pivotal year in Germany and for Berlin as a city. And I think globally, the last 2 years have been pivotal years for us as individuals in a nation and in a globe. And we are presented with new challenges. And if I just reflect on some of the things that came out through your presentations today, there was a lot of focus on health and well-being. There was a lot of focus on poverty, some focus on the systems involved in health, focus on mental health, focus on social inclusion during the pandemic, and of course things like sustainability and how we travel in a post-COVID world. So just some of it, a lot of the theming and the thinking I thought was reflective of, or could be sort of translated into the time in which we are living. And I just really wanted to just acknowledge that there are going to be some emerging themes that we see come out and beyond today from as a result of the last 2 years. So pleasing to see some of those themes come up through there. I want to also thanks to the jury. Special thanks to Jess and Megan and the team. Megan, Falling Walls is following you wherever you go. Hans, thank you always for your passion. You're an incredible champion of this opportunity, and of course, same with Professor Roseman as well, and to the Falling Walls Lab in Canberra and globally, to the Australian Academy of Science and to UQ. Well done for keeping this well and truly cemented in Queensland. And yep, look forward to seeing how your careers transpire and move forward through this period and into Canberra in a couple of months' time. So thank you.
Thank you, Shannon, for your enthusiastic comments about this competition. I mean, it is a competition, we need winners, but it's really about spreading the ideas as widely as possible. And we make the same observation, the ethical teams, we discussed that in our breakout when the jury was deliberating. They are really the foundation of many of the big changes that we need. And we also discussed that we need a link between the research and the commercial world to make this all work. And we need the biggest possible audience of decision makers. And that's what Falling Walls Berlin is all about. It's the audience. It's you out there to take the ideas that you heard of today and tell others. Come back, ask questions, find decision makers that can turn ideas into reality. So on that note, I would like to welcome you all back in September. For the details, please go to the webpage of the Academy of Science. The finale will be announced very soon, and you find the Zoom links, the Facebook, the YouTube, all the ways we communicate. Thank you for joining in today, and thank you in particular for all the presenters for taking on the challenge. So good luck to you, and see you back in Canberra. Bye.
Bye.